So the deepest point of our ocean is just here in the Mariana Trench, about 11,000 meters below the surface. It's pitch black, it's cold, but there is life there. Crustaceans scavenge on the seabeds, jellyfish and other weird gelatinous things drift above them, and there's even stuff living in the mud. But there's no fish. Fish have taken coral reefs, polar seas, and vast amounts of the deep ocean. And yet somewhere above the bottom, in fact precisely here, they stop. Above this line we find fish. Below it we find precisely none. This isn't a quirk of the Mariana Trench either. This boundary exists in every trench across the Pacific, thousands of kilometers apart. So some animals, like the crustaceans, cross this line and get to the very bottom. But the fish don't. Or maybe they can't. So to find out why, I spoke to the man probably most likely to know, Professor Alan Jameson. Now, Alan has spent his entire career exploring the deepest parts of the ocean. I mean, he's pretty much been to the bottom. But most importantly, he also led the expedition that filmed the deepest fish. So I asked him the obvious question. Why can't our fish get down here?
Everyone thinks it's about being crushed and all this kind of stuff. That's not it. It's kind of that, but it's at a cellular level.
So it isn't the weight of the water that's kind of flattening the fish. Something stranger is happening. And the answer to the question didn't come from an expedition to one of the world's deepest trenches. It came from a laboratory and a graph that nobody could explain.
Yes, the story of the deepest fish came around 2006, where I was an engineer by trade who was getting involved in all this deep sea biology stuff, and one day we had a bit of an epiphany as to why does everyone operate to 6,000 metres, why doesn't anyone go to 11? Because engineering wise, it doesn't take that much to push it that bit deeper. If you've gone to 6, you might as well go to 11, right? So for me, it was a case of like, wow, I want to build a camera that can explore a bit deeper, and we went through all the literature about what's known, and back in the 50s, the Danish pulled up a couple of snailfish from, I think, 7,500 metres, and they were pretty beat up. So I built this video camera and we went out to the Kermadec Trench off New Zealand. Day one, we put the camera down at 6,000 metres and we found this big prawn, which is something that apparently at that point shouldn't exist at those depths, but it did. Second one, we put it down, and I remember looking through the videos as it came back, and I just saw this tail just swish across the screen. I'm like, yeah, there is a fish here. Like, oh wow. That was the first time anyone ever recorded a fish at those depths, right? By 2008, we went up to Japan and we put it down to 7,700 and we filmed like 22 snailfish in one shot.
But filming 22 fish in one frame tells us something important, right? I mean, these fish aren't sort of clinging to the edge of existence down there. There are entire populations of them, feeding, breeding, at depths far greater than anyone had assumed. Which raised the real question. If fish were comfortable going this deep, how deep could they actually go? Well, at almost exactly the same time, someone on the other side of the world had found the first clue. Not in a trench though, but in a freezer full of mussel samples. This is Paul Yancey, a professor at Whitman College in Washington. So Paul studies something called osmolytes that are basically molecules that help a cell deal with their surroundings. One of those osmolytes is this thing, TMAO, or trimethyl 1,2,3-methyl amine nitrogen N oxide. And in deep sea fish, this thing right here does something pretty cool. Okay, so depth, concentration of TMAO. What they found was, the deeper the fish goes, the higher the concentration of TMAO. And it followed almost a perfectly straight line. Which is weird because in the ocean, pretty much nothing moves in a straight line. Temperature wanders, salinity wanders, but TMAO was tracking depth almost perfectly.
They extrapolated this line and said TMAO should reach a state of isosmosis, which is basically saturation point, at around maybe 8 to 8,400 metres, something in that range. So he's like, at that point, if all bony fish are using this molecule, which they all do, they should be able to get deeper than 8,000 metres plus or minus.
Now on paper this is a pretty bold claim, right? I mean, a hard depth ceiling for every fish in the ocean, at depths that no one's really sampled before. But there was one way to test it. Someone needed to bring back a fish from the very bottom of the line.
I spoke to him, I said, look, I think I'm the only guy in the world who can get you a fish from that end of your line. And he's like, nah. It's like, no, I swear, we've been filming them, right? We just need to build a trap. And we went off New Zealand, put the cameras down and put the traps down. And within 40 hours, we had about 6 or 7 snailfish on deck. So we took the sample and sent them all to Paul Yancey. And he was like, you're not going to believe this. They are absolutely bang on that extrapolated line.
So the prediction held. A line drawn in a laboratory had correctly guessed the chemistry of a fish from the deepest reaches of the ocean. But a confirmed line still doesn't really explain itself, because although we know where the fish stops, we still don't really know why. And to understand why, we've got to zoom in a little and look inside the cell. Right, this is a pretty weird way to explain this next bit, but it's all I can think of, so bear with me. Let's imagine that this camping chair is a protein. Now, proteins aren't sort of shapeless blobs that just drift around your cells. They're molecular powerhouses. Whilst they hold their shape, they can do their job. But like this chair, if they lose their shape, like so, anyway, if they lose their shape, nothing really changes, right? It's still a chair, but I can't sit in it. It no longer performs its function. And your proteins, right, unlike this chair, have thousands of jobs to do inside the cell. They're building tissue, digesting food, repairing DNA, but only whilst they hold their fold. Pressure doesn't crush the fish. I mean, there's no air spaces to collapse, which is why a specimen that's brought to the surface looks identical to one swimming around in the deep. The danger isn't to the fish's body, it's to the fold of the proteins. If you go too deep, they kind of lose their shape and do lots of weird stuff. Which is where TMAO comes in. We know that fish that live really deep have cells that are loaded with this because it helps their proteins hold their structure under immense pressure. Exactly how is still a little bit contested, but the effect is clear. More pressure requires much more of this. But there's a point just here where the line ends. So let's say that each one of these little balls is a TMAO molecule. So in shallow water fish, they need only a few. But at 4,000 meters, they need many more. And in the deepest trenches, they're pretty much full. Got it? But TMAO doesn't just study proteins, it also acts like a kind of salt. The more of it you add, the saltier the cell gets, until it's the same saltiness as the seawater around it. And the problem with that is that if it's as salty or saltier than the seawater around it, then water can enter the cell via osmosis, which definitely isn't a good thing. But all of this was just theoretical. We still hadn't found the deepest fish, the one that's swimming around right up against our red line. Finding it would come down to a variable that, up until now, everyone had missed. So for years, the deepest fish sat at about 8,100 meters in the Mariana Trench. It looked like the hard stop that the graph had promised. But then Alan brought in the one thing that the samples hadn't let him test.
Mariana snailfish became the deepest fish for a long time, until around 2022 I published this big census of all deep sea fish. And at that point, the thing we had never been able to factor in because we didn't have the volume of samples to do it is what about temperature? Because temperature and pressure have the same perturbing effect on the cell. So if it's warm, you can go deeper.
The cold sort of stiffens a cell like pressure does, so a fish in slightly warmer water has a little bit more room to spare. Which means that the deepest fish on Earth might not necessarily be in the deepest trench, it might be in a warmer one.
Then I was looking across the records of them all, thinking the Mariana snailfish is not the deepest fish. Because there's one trench in the world that has the right depth for there to be a deeper one, and is warmer. So I wrote in this paper, like, I reckon the deepest fish in the world is off Japan, and it's probably in the Ogasawara Trench. I got the chance to go there a couple of years later, and right enough, 8,336. And at that point, it was one juvenile, probably 3 or 4 inches long, that just swam past the camera once. It really was like the last bastion of fish. And so at that point, I think, like, all right, I mean, it's not worth chasing this anymore, right? This is exactly it.
So a graph drawn in a laboratory predicted the deepest fish, down to the very trench it would be hiding in. And let's be honest, this probably isn't what you thought it would look like, is it? I mean, first of all, it's kind of small.
The thing is with snailfish are not deep sea fish. Snailfish are shallow water. They're a shallow water family. There's 330 species. Most of them you can find in estuaries and rivers. What's cool about the Cuscales and the Grenadiers is they're the kind of fish you would look at and go, that's a deep sea fish. They've been around for years. They've nailed this whole deep sea stuff. And then there's this little new guy on the block, the snailfish, has come down and punched easily 1,000 meters deeper than they have. And they just look like stupid little tadpoles, right?
No, don't listen to him.
They're just like, no.
The fish that got the deepest isn't a sort of grizzled veteran of the deep. It's a fish from a family that you're more likely to meet in a river mouth.
They don't have a swim bladder, they don't have scales, they're just covered in gel, they're practically transparent. They don't have big teeth because they use suction feeding. They kind of hang around in what appears to be social groups. They're not big nomadic wanderers that come in and destroy some bait and then just leave on their own. If we went down the street with a microphone and asked people, what do you think the deepest fish in the world looks like? They'd probably come up with black and lures and big teeth and big beady abs and all this kind of stuff. And you're like, that's this.
But despite its mastery of the deep ocean, our snailfish is still leaving about 3,000 metres of water on the table. Plenty of other animals have made it down here, but not the snailfish. Why? So Alan has recorded hundreds of days of footage south of this line, but he's never seen a fish down there. In the deep, there are only really 3 groups of fish, and each of them has its ceiling much higher up.
You've got the jawless fish, which is your lampreys and hagfish. They mostly stop at around 1,800 metres. There's a couple of hagfish that will go to 3, but that's it. They just don't go any deeper than that. Then you've got your chondrichthyes, which is your sharks, rays and chimeras. They've got cartilage for bone. They generally don't go beyond 3,500, and that's partly to do with, they use TMAO as well, but they mix it with urea, and the combination of TMAO and urea won't get you past 3,500. Same laws apply. And the other problem with those sharks, rays and chimeras is they use their liver for buoyancy. They don't have swim bladders. And when you get down into colder and colder environments, when you're using oil as buoyancy, oil becomes sticky and weird, and maybe there are other factors at play. In nature, you find that there's not normally exclusively one reason for something. It's normally an interplay of a few things.
So every group of fish hits a wall, and each one of them is higher than the snailfish is. But if you look beyond the fish, life splits cleanly into 2 groups.
There's those that go all the way. So you've got your amphipods, isopods, gastropods, polychaetes, all these things. They will go 11,000 metres and don't care. But then you've got brittle stars, sea stars, crabs, lobsters, fish, scaphopods, all these things. They all go to about 8,000 and they stop. So it turns out they're using a whole cocktail of different osmolytes. Things like 5 or 6 all interacting with each other and stuff. And so that became, clearly they're doing something different and something much more complex, and there might be things in there which are a byproduct of something else. But then we started looking at things like prawns and stuff like that, and it turns out they only use TMAO as well. So regardless of how different the animal is, the pressure stabilizing molecules in their cells are the same. So that's why many groups are only going to 8,000 metres.
So the chemistry that enables our friendly snailfish to get down to 8,000 metres is the same chemistry that stops it there. So then Alan asked the question, why there?
So I started thinking, well, over the last 20, 30 million years, why would any animal in the sea, or lots of different animal groups in the sea, evolve a solution that only gets you to 8 and not 11? Because, like the engineering analogy is, if you're going to do all that work to get that way, why not go that last 2% or that last 3,000 metres? The only places deeper than that are trenches, which are tectonic, and they're constantly moving and being subducted and stuff. And then we're thinking, well, maybe 8,000 metres is an old, foolish depth.
So this is where the story stops being about the ocean and starts being about the planet. We kind of assume that the ocean has always looked the way it does today, but it hasn't. So the deepest trenches on Earth exist in places where plates like this one grind beneath another one. The plates are young, so they move, and they tear the seafloor open far below the level it used to sit at. So maybe 8,000 metres isn't arbitrary. Maybe it's a memory, the depth that these fish managed to reach before the ocean did this.
And all the animals that don't care about depth just fill it because they don't care. And everything else went, no, we've evolved to this point and now there's a deeper point, but whatever, okay.
But there's one thing that might be able to move our line: temperature. If you remember, colder water means fish have to sit shallower, but warmer water buys fish depth.
So, for example, like a 5-degree change in water temperature, what does that mean in terms of depth distribution?
They reckon a change of 2 degrees Celsius equaled 1,000 metre depth range.
Wow.
So the invisible line that fish can't cross has never really stayed still. In fact, it kind of drifts up and down as the Earth changes. And it's moving now. And maybe at some point in the future, our small jelly tadpole-like thing might just make it to the bottom. By the way, guys, if you're new here, I do actually do some of my own deep water exploration over on my main channel. I also have a Patreon for people that want to see all the footage as soon as it's shot and also help support these deep sea adventures and all the equipment that we have to use. If you enjoyed this video and feel so inspired to chip in a few dollars each month just to help out, that would be massively appreciated. But yeah, plenty more to come, loads of equipment being built, tons of stuff to show you. Really hope you enjoyed this video, and I'll see you on the next one.